Hi, my name is Amy Ryan, and I'm an assistant professor of medicine at the University of Southern California. Today, I'm going to share with you a presentation titled Cellular Targets for Successful Gene Editing and Cell-Based Therapies for Durable Correction of CF Lung Disease. I have no disclosures. So when we're thinking of regenerating a CFTR expressing airway epithelium, we need to think about what the options are. And these include gene editing of endogenous stem cells, and autologous cell therapy using either ex vivo expanded airway stem cells or iPSC-derived airway stem cells. And while we have these cells, we need to decide, well, really, what are the best targets for airway regeneration in cystic fibrosis? We need to be targeting a stem cell that can self-renew and reside in the airways for a long period of time, that they are capable of differentiating and regenerating all the cells that give rise to the airway epithelium, and that they can differentiate in cells that are capable of conducting chloride via the CFTR. Specifically for gene editing, the cells must be accessible by either the virus or the gene editing technology that is being delivered. And for autologous cell therapy, we really need the cells to be able to be expanded and maintain stemness ex vivo, ready for engraftment into the airways. This is a beautiful diagram from Dr. Engelhardt, where you can see the full range of the tracheobronchial tree going from the proximal distal axis. And along this axis, you can see the different stem cell niches highlighted and the stem cells that reside in those niches. In the proximal conducting airways, we have the submucosal glands, of which they comprise of cells, myoepithelial cells, which are known to be able to replenish the surface airway epithelium under severe injury. In the same region as the airways and the proximal conducting airways, we know that the basal cells are able to give rise to Klebsiella ciliated and goblet cells comprising of the mucociliary epithelium. As you move more distally, there are a number of other stem cells, such as variant Klebs cells, bronchiolar alveolar stem cells, and alveolar type 2 stem cells. For the sake of today's talk, we're going to focus and hone in on the basal stem cells, because I believe that the cell-based therapies for cystic fibrosis are going to need to focus on the conducting airway epithelium and restoration of mucociliary clearance, as a conducting airway is the primary site for mucus production, for infection, and for inflammation, and particularly in cystic fibrosis. But we know that the epithelium is relatively quiescent under homeostasis, but upon injury, the basal cells can rapidly self-renew and differentiate and regenerate that airway epithelium. And the basal cells are perhaps the most predominant and best characterized stem cell in the cartilaginous airways. And this is a diagram from a work done by Bridget Hogan that highlights the classical markers of the basal stem cell, which are keratin 5 and P63 and keratin 14. These cells are able to cycle and self-renew, and they're also able to differentiate and give rise to a multipotent progenitor, which increases in its expression of keratin 8, designating a lumen or faded basal cell capable of differentiating two neuroendocrine, ciliated, and secretory cells of the conducting airways. So how do we get basal stem cells? How do we expand them, and how do we gene correct them to deliver them back as an autologous cell therapy? Well, we can obtain them from living donors using minimally invasive techniques such as collection of induced sputum samples, bronchial lavages, and endobronchial brushings or biopsies. However, these only generate a small number of cells, somewhere in the range of 2 × 10⁻³ to 2 × 10⁻⁶ cells. Thus, the paucity of bronchial epithelial cells poses a considerable challenge when we're thinking of autologous cell therapy. We need to be able to successfully expand these cells ex vivo to much larger numbers while maintaining their stemness. Recent work from Scott Randell's lab has shown that ex vivo expansion of cells from freshly isolated day 1 passage 1 cells due to fully passage P1, P3, and P5 cells, there's a depletion of the NGFR and EpCAM expressing basal stem cell over time. Scott's lab has used this conditionally reprogrammed culture technique, which the cells are grown in specialized media on NIH3T3 cells. And what you can see by passaging the cells using this technique, you can see a substantial increase in the number of NGFR-expressing basal stem cells that are maintained over time. This is summarized in the graph here again, where you can see a significantly increased number of NGFR-expressing basal stem cells over time. In addition, the CRC culture technique, Scott also showed that it's able to increase the number of differentiated ionocytes from these cells. And as we know, the ionocytes have been identified as being a hub of CFTR activity. So being able to maintain a stem cell that is able to successfully give rise to ionocytes could have a significantly better impact as a cellular therapy. Data from my own lab and that of the CF Foundation in collaboration with Jed Mahoney was shown that ex vivo expansion of of human bronchiopithelial cells or basal stem cells decreases the regenerative potential over time. So here you can see air liquid interface cultures from passage 1 cells and from cells that have been passaged 4 times in bronchial life media. And you can see that after 4 passages, this particular line of patient-derived cells has a significant reduction in differentiation of ciliated, goblet, and secretory cells, and also a breakdown in the efficient generation of an epithelial barrier. We've actually sequenced cells that have been passaged over a number of passages, from passage 1 to P4, grown in Begum, the bronchial epithelial growth media, and also in media that's been supplemented with dual SMAD inhibitors that were found by Jay Rajagopal's lab to be able to expand the regenerative basal stem cell, and also those grown in pneumocult EX+. And what you can see that is that the P1 cells cluster away from the passage cells, and also that the Begum passage cells cluster away from the dual SMAD and the pneumocult EX+ expanded cells. One thing that we see over time is that there's significant upregulation and downregulation of a number of genes. And when you look at the pathways that are most impacted over time, you can see that these are associated with cell cycle and DNA replication, therefore indicating that this is a significant, these pathways have a significant impact on cells as they expand ex vivo. One thing that can happen when you expand cells ex vivo, whichever cell it is that you have clonal selection for those that thrive in the culture environment that you have in your dish. And we do now know that there are subpopulations of basal stem cells, work done by a group in the Cystic Fibrosis Consortium on Stem Cells, published recently that there are 5 transcriptomically independent subsets of basal cells, as can be seen here, and they've shown the differential expression of several genes associated with each of those subsets. And within those subsets, they've also identified cell surface markers that could be used to separate and purify out the basal cells to be able to functionally characterize those basal stem cells. Here is one example where we've used CD66 and CD266 to separate out one of those subpopulations, the basal 1 population, which is the population that is thought to reflect the basal stem cell based on the marker expressions that we know. However, one thing that was interesting in this study was that they compared cystic fibrosis patient-derived basal cells to non-CF patient-derived basal cells. And there is actually a significant difference in genes that are expressed in the different populations between CF and non-CF. And moving forward, I think one of the priorities going to have to be is to understand what changes occur in basal stem cells in cystic fibrosis, and does this impact the regenerative capacity and long-term stemness? Because these are going to be important factors moving forward as we consider both gene editing, gene therapy, and autologous cell therapies in CF patients. So as I just said, is one of these basal cell subpopulations going to be a better target for editing and cell-based therapies? It's questions that remain to be answered based on the data that's been shown. Can these stem and progenitor stem cells be clonally enriched? So can we use those cell surface markers to select for them? Should we be targeting specific clones for gene correction, or should we be targeting all basal cells? And I think important in CF has just shown that by the transcriptomic differences in CF and non-CF basal cells is that how do changes in the stem cell niche in cystic fibrosis impact the different clones? And if cellular subtypes develop as a product of their environment, then clones will or should exhibit plasticity based on the culture conditions and the environment. And this information could be used to both manipulate the in vivo environment to be more conducive and receptive to maintaining stemness of the autologously transplanted cells. Also, they could be used to improve the ex vivo conditions for expanding these cells so we maintain a better quality of basal stem cell. And can we utilize these niche and stem cell interactions to be able to optimize strategies to facilitate cell engraftment or to better target endogenous stem cells? So as we've mentioned niche, our lab has done a little bit of work looking at the niche and how it may be impacting airway regeneration. Cell therapy or stimulation of endogenous airway stem cells and targeted gene correction are all likely to occur in a CF lung that has already undergone some changes, at least in the stem cell microenvironment. Here's an image of a CF lung where we can see that there is some basal cell hyperplasia in this region of the airways. We've got a big mucus plug blocking the airway lumen, and we've got excessive mucus within the submucosal glands. And we can see a huge amount of inflammation here and infiltration of immune cells. All of these are going to or possibly have an impact on airway stem cells and their capacity for regeneration. So we need to think about things such as changes in the extracellular matrix, in the inflammatory cells that are present in proximity to the airway epithelium, and the epithelium cellular composition, how these are going to impact airway stem cells. We also need to consider how do the genetic, epigenetic, and proteomic changes in the CF lung impact the niche and the regeneration. So do transcriptomic data predict functional changes in airway regeneration? Do regional specific changes in the cellular niche impact the airway stem cell function? And I think all of these questions have been posed by the field, but still remain to be answered. We've looked a little bit at how immune cells can impact stem cell differentiation by doing some in vitro studies, looking at basal cell differentiation to the mucociliary airway epithelium. We developed this contact model where macrophages are co-cultured with the epithelium on the basolateral side of the insert. And over time, we can see that we're able to see differentiate epithelium. And on the underside, we can see these large flattened CD11b expressing macrophages. When we compare wild-type macrophages to CF macrophages, we can actually see that in the wild-type epithelium here. So we basically just changed the mutation in the macrophages, but not the epithelium. In this case, we can see that there's actually a significant change in the speed at which the cells are able to differentiate into multiciliated cells. And that wild-type macrophages actually stimulate differentiation to multiciliated cells, whereas there is a decrease in the efficiency of generating multiciliated cells when the CF macrophages are present in close contact with the epithelium. Another thing we also observed was that there was an increase in macrophage migration with the CF macrophages through the epithelial layers, can be seen by the macrophages protruding through the epithelium as indicated by these red arrows. We also see functional differences in the macrophages such as phagocytosis, and all of these seem to have an impact on the ability of the basal stem cells to function and differentiate. These are going to be interesting studies for us to follow up on. Another part of this, as I mentioned, if we're thinking of autologous cell therapy, expansion of endogenous stem cells is one option, but differentiation of patient-specific iPS cells into lung basal stem cells is another option for cell therapy, where we take the patient cells, reprogram them, we can genetically correct these, clonally expand those, and differentiate them into cells to be able to give these back as an autologous cell therapy to the patients. We can also use these cells for mechanistic research and high throughput screening to develop new precision medicine approaches for those patients too. So iPSC differentiation has evolved over the last 10 years. Here is our current differentiation protocol going from pluripotency through definitive endoderm, anterior foregut endoderm to NKX 2.1 expressing lung progenitors. We've now modified our protocol to be able to selectively expand basal cells that express NGFR and that are capable of differentiation at the ALI to give rise to a mucociliary epithelium. So we basically select out our NGFR expressing cells. We can grow these in the conditionally reprogrammed media that Scott Randell developed and to Reynolds. And we can see that we're able to generate iPSC-derived basal cells expressing P63 and keratin 5, and they're able to grow in the different commercially available medias. Finn Hawkins has also developed techniques, and he recently published his methods in cell stem cell, where he's able to grow his iPSC-derived basal cells in 3D spheroids. He's able to successfully expand these, sort these, and again plate them and differentiate them at the ALI. And he also sorts for NGFR and EpCAM, and he has a reporter line that is able to validate that these cells are also expressing P63. He can use the reporter line for sorting of these cells and dissociation of the basal stem cells. However, the iPSC-derived basal cells do have a tendency to fail over longer periods of time at the air-liquid interface. Our 2D expanded basal cells are able to generate TIR, but after about 2 weeks of differentiation at the ALI, that barrier starts to break down and the quality of the epithelium then deplenishes. And in the 3D expanded basal cells, we get a better quality of airway and more consistent airway epithelium. However, again, by day 21, we see this barrier starting to break down and the differentiation of the cells not holding up as a good quality differentiate epithelium. This data here is shared with us by Jed Mahoney from the CF Foundation. So this indicates that the iPSC protocols, although we get very similar basal cells to endogenous basal cells, that there are still some differences, and we are currently working on identifying what those differences are and how impactful they are going to be when we're thinking of autologous cell therapies. So again, just summarizing the advantages and disadvantages for iPSC models. The disadvantages really are that, we still have uncertain reproducibility and differentiation in the physiological responses when we look at CFTR activity. I didn't show you that data. The protocols are lengthy to mimic the human development, and we do see transcriptomic and phenotypic differences between iPSC-derived and primary cells that we need to really fully evaluate to see how significant these are when it comes to autologous cell therapy. I spent all this time talking about basal cells. We also know that myoepithelial stem cells are able to fully differentiate and give rise to all of the cells in the epithelium. This was beautiful work done by John Engelhardt's group, where he showed that cells could, the myoepithelial cells could come out of the submucosal glands and replenish the surface airway epithelium only upon severe injury. And he demonstrated this using a naphthalene injury model of the airways, where you can see GFP expressing myoepithelial cells that are residing in the epithelium and co-expressing keratin 5. So these myoepithelial cells are able to give rise to the multipotent stem cells of the surface airway epithelium, and these could be another potential target for gene editing and for cell therapy when we're thinking of correcting the CFTR. So in conclusion, basal cells are the predominant stem cell in the conducting airway, and we have patient-specific but limited these are patient-specific but have limited ex vivo expansion without clonal selection. Physiological relevance of subpopulations is currently unknown, and accessibility of basal cells for gene editing may be challenging without injury. Exogenous expanded or iPSC-derived and gene-corrected basal cells could be used for autologous cell therapy. However, we're still not sure about what the impact of the niche on function is going to be in CF and whether changes in CF basal cells are going to have a significant impact on this. I just wanted to say thank you to the people in my lab, Yana Narworth, Ben Calvert, Eric Quiros, Shana Bush, and Rosemary Cease, who contributed to this research, and to our collaborators.
Hi, I want to thank Paul and Hillary for inviting me to talk today and all the organizers for coordinating this conference. Today, I'll discuss with you recent work in the Lu Lab using base editing and prime editing towards the treatment of genetic disease. Here's my disclosure slide. My only disclosure is that I'm co-inventor on some genome editing patents filed by the Broad Institute in Harvard. There are many kinds of genomic mutations that can cause disease in humans. Single nucleotide variants make up about half of all these disease-associated mutations. They're denoted by the blue and red pie slices here. Cas9 nuclease has revolutionized our ability to modify cellular genomes, but it's not well suited to efficiently correct single nucleotide variants. Following the introduction of a double-stranded DNA break in the genome by Cas9 or another nuclease, cellular DNA repair machinery typically rejoins these two free ends with end-joining processes. This often results in insertions and deletions, or indels, in the resulting alleles. It is possible to provide a donor DNA template and encourage cells to use homology-directed repair to rejoin those strands, resulting in a precise and programmed editing outcome, like a single nucleotide correction. However, this process is typically inefficient in slowly dividing or non-dividing cells, as make up most of the human body. Furthermore, alleles that do not receive the intended edit typically receive an indel instead. In February of this year, there were 43 ongoing clinical trials registered using CRISPR technology. Most of them were designed to disrupt target alleles, and none of them were designed for precise nucleotide modification. To address this problem, my predecessors and colleagues in the Lu Lab developed base editors, which efficiently introduce single base edits without producing double-strand DNA breaks. And therefore, they lead to purer editing outcomes with few indels. The C to T base editor was first developed, then the A to G base editor, and recently a C to T editor that works in mitochondria. Base editors, the A base editor that is, or ABEs, are particularly exciting to me because they can be used to correct nearly half of all single nucleotide variants that cause disease in humans. ABEs work by deaminating adenosine bases, creating inosine, which is as read as a G by cellular polymerases. Here's a diagram of the ABE domains in that A base editing process. ABE is composed of a Cas9 nickase, so this enzyme can no longer introduce double-stranded DNA breaks into the cell, but only nicks one strand, and that's fused to a deoxyadenosine deaminase. When this construct binds to genomic DNA at its target site, defined by a 20-nucleotide sequence stretch in the guide RNA, it releases an R loop of free genomic DNA displaced by annealing of the guide. And a small window of nucleotides within that R loop is available for base editing by the deaminase. Following activity of the ABE, an inosine is now encoded in the DNA, and there's a nick on the opposite strand that's not edited. This nick directs DNA repair so that that non-edited strand is replaced based on the template of the edited strand, converting now both strands to the desired edit. I aim to apply ABE to modify the sickle cell disease mutation towards a more precise editing outcome that efficiently eliminates the disease-causing mutation while introducing a healthy allele. Sickle cell disease is the most common genetic disorder that's deadly and is caused by an A to T mutation in the adult beta globin gene, leading to this glutamate to valine mutation. This leads to polymerization of deoxygenated hemoglobin and a lot of pain in patients and early death. Most patients do not have a matched donor for allogeneic bone marrow transplantation, and so many ongoing efforts, including ours, aim to modify a patient's own cells so they can be retransplanted back into the patient and cure the disease without any risk of graft versus host disease. We used an A base editor containing a specifically evolved Cas9 domain that recognizes a non-canonical PAM, since NGG PAM and other standard PAM sequences are lacking near the target mutation. This allowed us to place that target nucleotide, the causative one for the sickle cell disease, right at position 7 of our protospacer, which is well within the editing window. Potential bystander edits with this editing strategy include positions 9, 12, and 16, but only the edit at position 16 would lead to a coding change. We electroporated this editor as an mRNA or RNP into hematopoietic stem and progenitor cells from sickle cell patients, and we reached up to 80% editing of the sickle cell mutation. We saw no editing at position 16 that would lead to non-synonymous editing outcomes. It would be indicated by a red bar here. And we saw less than 3% of other undesired outcomes like indels. In order to determine whether this editing strategy really modified the long-term and bone marrow repopulating hematopoietic stem cells, we assessed their stability in mice following transplantation. So after we electroporated our ABE mRNA, we transplanted cells into immunocompromised MBSGW mice and waited 16 weeks before harvesting bone marrow for analysis. The idea being that if we modified cells that had already started to differentiate, then they would be lost during this course of the 16 weeks and only stem cells would remain at the end. What we observed was that in all lineages after the 16-week period, the allele frequency of the desired editing outcome was around 68%. Using single-cell RNA-seq, we defined these fractions of those cells that received a biallelic edit in both copies of the beta-globin allele or a monoallelic edit in only one of the copies. And it was about 50-50 between those two pools. In total, we edited between 80 and 90% of all of the cells with at least one allele. And we'd expect that any of these cells receiving even just one edit would be free of sickle cell disease. Using HPLC to quantify the hemoglobin outcome, we see about the expected ratio of around 68% of the sickle globin that had been converted to the edited globin called Macassar globin, or beta G, here denoted in blue. And when we put cells, the cells that were extracted from these mice under hypoxic conditions to induce cell sickling, we saw that the propensity to sickle in the edited cells is much, much lower, about 5-fold reduced sickling. This mouse model was reassuring to us that we were really editing the long-term hematopoietic stem cells from human patients. But because human cells are short-lived in the mouse circulation, in the peripheral blood system of the mouse, we weren't able to measure hematological defects and any restoration that's granted by our editing strategy. Therefore, we turned to a mouse model called the Townes mouse, which contains a human beta-globin locus. And there are Towne mouse derivatives that contain the sickle cell mutation and are homozygous for that sickle cell mutation. And these mice are very sick with sickle cell-like symptoms. There's also a related mouse model that contains one healthy adult beta-globin allele called HBBA. And when you have a heterozygous mouse with one healthy allele, one sickle allele, these mice have sickle trait, which is largely a healthy condition. These were used as positive controls in an experiment where we assessed whether editing and retransplantation of these mouse hematopoietic stem and progenitor cells could correct disease phenotypes. So we edited cells from the homozygous sickle mouse and transplanted these into one mouse population. We also transplanted unedited sickle cells as a negative control and transplanted healthy cells as a positive control. After 16 weeks, we sacrificed these mice and assessed their hematological parameters. We saw that editing of about 50% was achieved in these mouse cells and was not significantly changed, though somewhat decreased over the 16 weeks during transplantation. The fraction of beta-globin protein that was measured by HPLC was between 70 to 80% at all time points. We think that this is in excess of the editing frequency because cells that have been edited are now healthier and they're longer lasting in the periphery. And so those edited cells now make up a larger proportion of the blood. Strikingly, we observed that in all of the parameters we measure in a complete blood count, the hemoglobin, the total hemoglobin level, the percentage of reticulocytes, the red blood cell count, and the white blood cell count, each of these parameters is restored to be statistically similar to the healthy controls and statistically different from the unedited controls, which have a disease phenotype. We placed blood from these mice under hypoxia to examine sickling phenotypes. We saw that unedited sickle cells, unedited cells that contain two sickle globin alleles, readily sickled under hypoxia. The cells that were edited, transplanted into mice, and then extracted after 16 weeks, largely did not sickle. This was you can pick out several cells that do sickle, but on average, this blood sample looks much more similar to the positive control of a healthy cells where no sickling occurs. We conducted colony-forming assay to look at the fraction of cells that were edited with one edit, two edits, or were entirely unedited. And similar to before, we see approximately equal ratios of cells with monoallelic or biallelic edits. In this case, it's around 80% of cells that contain at least one allele. We also looked at the splenic pathology of these mice. Mice that were homozygous for the sickle cell mutation contain this characteristic expansion of the red pulp, which signifies extramedullary erythropoiesis, as the spleen is now being used as a site to produce more blood cells because of the high turnover in this mouse model. However, some animals that received our edited cells had a much more wild type looking spleen compared to this when compared to this positive healthy control. Likewise, the enlarged spleen phenotype, splenomegaly, that's characteristic of this mouse model, was largely corrected in mice that received our edited cell population. So this quantifies the correction here in the total spleen mass. We achieve about 60% correction back to a healthy state. Shifting gears into new work that was conducted in collaboration with Paul McRae's lab, we looked at cell models to treat cystic fibrosis. This work's currently in press in nucleic acids research. Here's the DNA sequence encoding the 553 stop codon CFTR mutation that's currently intractable to cystic fibrosis small molecule therapeutics. We used a CUFI3 cell line that's heterozygous for this R553X mutation to assess editing and correction of the cystic fibrosis phenotypes in these epithelial cells. We electroporated an RNP of an Abe editor and saw restoration of the wild type sequence from 50% in this heterozygote cell line up to 90%. In primary cells, these are primary human airway epithelial cells that are heterozygous for the 1282 stop codon mutation. We conducted a similar protocol electroporating Abe RNPs, and we observed that the frequency of that wild type allele increased from 50% to around 70%. Using an Ipsc chamber to conduct analysis of the conductance of these cells, we observed that both of these cell lines, both at the R553X and the W1282X, showed a marked improvement in the conductance, and they're much more similar to a wild type cell now than they are to their original mutant cells before editing. So we hope to develop methods that we can efficiently deliver these editors into animal models towards applying these to treat patients. Base editors can correct the largest individual pie slice of human disease mutations, but there are many other terrible diseases that are caused by other kinds of mutations, not least of which the 508del CFTR allele, that's the most common disease-causing one in cystic fibrosis. In order to enable more efficient editing in these models, my lab mate Andrew developed a new strategy called prime editing. The prime editor is composed of a fusion of Cas9 nickase to a reverse transcriptase. Following nicking by the Cas9 nickase, the R loop can anneal to the complementary sequence we attach to the end of a guide RNA, and reverse transcriptase can write the desired sequences directly into the genome. The end result of this is a flap equilibrium where you have your newly installed edited sequence shown here following the red nucleotides, and the flap of endogenous sequence that's competing to hybridize with the unedited DNA strand. We believe that the cellular nucleases favor the excision of 5' flaps, which is helping our editing efficiency here. In a PE3 strategy, a second nick is introduced downstream, which causes DNA repair to replace the unedited strand and copy over that edited strand to increase the efficiency of the final edited product. And so using prime editing, we can directly replace any desired nucleotides with other desired nucleotides, or we can introduce deletions or insertions by adding extra or less sequence at those target sites. One advantage of this approach is that a single guide RNA and editor can be used to correct any number of possible mutations back to a wild-type genotype within a short stretch of DNA. Here we're showing a region of the CDKL5 gene, which causes an epilepsy phenotype in patients. And it has a cluster of possible disease-causing mutations all occurring right nearby each other. But a single editing strategy can correct all of them with a similar editing efficiency. Here I'm showing two different pegRNAs, pegRNA1 in this first cluster and pegRNA2 over here. And it's able to correct any of these sorts of mutations with equal efficiency. My lab mates, James and Peyton, recently identified that 3' nuclease activity degrading our prime editor guides, or pegRNAs, is a limitation for prime editing efficiency. They screened through several possible stabilizing motifs to protect the 3' ends from degradation, including these here called Evo pre-Q1 and MP not. They observed that both of these motifs work well to protect against degradation and increase the lifetime of pegRNAs and also increase prime editing efficiency at each of 3 different sites we've tested here in 3 different cell lines. So each set of 3 colored bars here indicates one target edit, and the first of these 3 triplets is using a standard unmodified pegRNA. The second uses Evo pre-Q1 fused to the end, and the third fuses MP not to the end. We see that these motifs yield around a 1.5 to 4-fold improvement in editing outcomes at these different cells and target edits. They call these stabilized pegRNAs engineered or ePegRNAs, and this work is now in press at Nature Biotechnology. In a separate study, my lab mate Peter Chen, in collaboration with Jeff Hussman in the lab of Britt Adamson, conducted a CRISPR I screen called RepairSeq. This knocked down each DNA repair protein in a pooled screen to learn which of them positively or negatively impacted prime editor activity. They identified that mismatch repair genes, mut-S and mut-L in particular, when disrupted, cause a much higher prime editing efficiency. Peter then devised a strategy to inhibit mismatch repair by delivering an engineered dominant negative mutant of the MLH1 gene. Here's a table now showing the different components involved in each of these different prime editor strategies, just so I can most accurately describe the nomenclature. So PE2 strategies use just the prime editor itself and the pegRNA directing the enzyme and encoding the desired edit. PE3 editing strategy also includes a nicking guide RNA that nicks that non-edited strand and helps direct DNA repair to install the edit. What's new in this study in press now is PE4, which is akin to PE2, but also includes MLH1 dominant negative, and also the PE5 strategy, which is akin to PE3, but includes this MLH1 dominant negative. PE4 increases the editing efficiency by 1.5 to 3-fold over PE2. PE5 often has just a slight increase in editing efficiency over PE3, but we see in each case that the indels shown in the checkered bars here are markedly decreased when using MLH1. So we think that by inhibiting mismatch repair, we're increasing the total amount of desired edits that we're getting at the same time as decreasing the number of alleles that turn out as indels. All of these recent improvements to prime editing synergize to even further increase prime editing efficiency. A new gene that we call PE-Max was developed by Peter by optimizing the NLS architecture and also codon usage in prime editors. Including the mismatch repair inhibition with PE4 substantially increases the prime editing efficiency, and using an ePegRNA even further increases that efficiency. And here, each of these dots indicates one of the 7 substitution edits that's tested at 7 different endogenous loci. This set of bars at the right includes that nicking guide. So both the editing efficiency and the indels are higher with these prime editing strategies. But we again see the same thing, that including the optimized prime editor gene increases editing efficiency, including MLH1 increases it further, and including an EPEG RNA gives a further boost to editing efficiency. We saw that these improvements also increase editing efficiency in clinically relevant cell types, bringing the editing up to around 30 to 60% in primary T cells when you use PE5. And in patient-derived human iPS cells, we get around 35% editing efficiency using this PE5 vector. So we hope that we'll be able to make use of all of these improvements to enable many other therapeutic edits, including those that cause cystic fibrosis. In summary, I showed that base editors can efficiently modify the sickle cell disease causing mutation and correct sickle cell disease phenotypes in mice. I showed that we can edit cell models of cystic fibrosis to restore CFTR function. And I showed that the new improvements to prime editing substantially improve its editing efficiency. So here in my acknowledgement slide, I really want to say a big thank you to my mentor, David, who supervised all of these studies and many more, and to all of our many coworkers and collaborating laboratories who made this work possible. It's been a pleasure to work with all of these people. And I also like to thank our funding sources here listed at the bottom. And I'm happy to take any questions. Thank you.
Hi, everyone. My name is Phil Borden, and I'm the Associate Director of Gene Editing at LifeEdit Therapeutics. And today I'd like to talk to you about some of our novel base editors and how they can be used to correct nonsense mutations in the CFTR gene. First, these are my disclosures. So I don't think I have to go into too much detail on this slide given the audience, but I'd just like to point out that for many patients who have CFTR mutations that affect things such as protein folding, ion channel function, protein maturation, or protein stability, there are several different therapies in the form of several potential therapies in the form of small molecules or antisense oligonucleotides, some of which are quite effective. However, for patients who have class 1 mutations in which no full-length CFTR protein is produced, gene editing really offers the only effective approach to rescuing protein synthesis and being able to help these patients with these mutations. So during my talk today, I'm going to be discussing a lot about base editing. So I thought I'd give a brief introduction about base editing in general. The most commonly used base editors come in 2 flavors: cytosine base editors and adenine base editors. And in both cases, these systems are composed of a partially catalytically inactivated RNA-guided DNA nuclease, typically SPY-Cas9. This nuclease functions as an RNA-guided targeting module that allows the base editor to target a specific region of the genome. Tethered to this nuclease is either a cytidine deaminase or an adenine deaminase. And this is really the chemical effector portion of the molecule that actually performs the specific base edit of interest. Other parts of the system are a small guide RNA, whose spacer sequence is complementary to a target region within the genome. Directly adjacent to that target region is an important DNA sequence called the protospacer adjacent motif, or PAM. And the PAM sequence is absolutely required for base editor function. And for example, with SPY-Cas9, its PAM sequence is NGG. Both C base editors and A base editors can perform very precise DNA edits. So for example, C base editors can convert Cs to T, and they do this by deaminating cytosine into uracil, which is then corrected to thymine. In the case of A base editors that can convert an A to a G, the adenine is first deaminated into an inosine, which then gets repaired as a guanine. And in both cases, these systems are able to affect single base pair changes, importantly, without doing any double strand cutting. This is very important because this prevents the formation of undesirable editing products, which could be deleterious to cells. Now, A base editors have been used specifically to correct nonsense mutations. So if you target an A that it's in the 2nd or 3rd position of a stop codon, those A's can be converted to a G, and the stop codon is then read through as a tryptophan. Alternatively, you could target the A on the opposite strand, and the cognate T in the first position of the stop codon is then converted to a C, which is read through as a glutamine or an arginine. This approach has been used to correct nonsense mutations in, for example, the XPC gene. So in this case, the target A was able to be converted to a G and roughly 10% of the time. And while this rate of editing seems rather low, it was significant enough to boost levels of full-length XPC protein to levels that were significantly higher than cells that were treated with either altilurin or gentamicin, which are two small molecules that are known to promote read-through of stop codons at a low level. A-base editors have also been used to target CFTR mutations. So in this recent report, a variant of SpyCas9 that has a single NG PAM tethered to a first-generation A-base editor was targeted using guides to the R553 stop or the W1282 stop mutations. And in both cases, these A-base editors were able to affect a large conversion of A's to G's at the target location. And in both cases, this resulted in the production of full-length CFTR protein. And in human bronchial epithelial cells, it was shown that this increase in full-length CFTR was able to partially rescue some of the deficits in CFTR function when the cells were treated with forskolin and IBMX. Now, unfortunately, not all CFTR mutations are actually able to be targeted by Cas9 and its variants due to their PAM specificity. So to illustrate this, in this paper, the authors looked at all of the possible CFTR mutations present in the CFTR2 database. And they found that only about a third of those mutations are actually addressable using an A-base editor. And of those targets, only an even smaller portion of those mutations are actually able to be targeted by wild-type Cas9 with its canonical NGG PAM. Now, of course, there are engineered versions of Cas9 that have more relaxed PAM specificities, such as NG or NGN. However, there are a couple of caveats to keep in mind with these engineered nuclease variants that I'll talk about in a couple of slides. In addition to the PAM requirements that are imposed by the nuclease itself, the adenine deaminase portion of the A-base editor comes with its own specific target requirements that further limit the number of mutations that can be targeted by an A-base editor. So, for example, when you target when you tether an adenine deaminase to an RNA-guided nuclease, there's a certain distance between the target A and the PAM that is required that sets a specific editing window. In addition to that, the target A must be on the correct strand with respect to the PAM sequence. And finally, the nucleotides that are surrounding the target A are of importance because the various adenine deaminases are known to have biases in the nucleotide context surrounding the target A that are known to affect their editing efficiency. So for example, if you look at the E60 or Y122 stop mutations, in both cases, the target A's lie too close to a canonical NGG PAM. So these A's are not within the editing window. They would not be efficiently edited. Similarly, if you look at the R1162 stop mutation, there's no canonical and ideal canonical Cas9 PAM near the target A. And so again, this A is not easily targetable. As I mentioned, there are engineered versions of Cas9 that have relaxed PAM specificities. However, there are a couple of caveats to consider with those systems. So to illustrate that, I'm showing data from David Liu's lab where they developed the current generation of A-base editors, the AB8 series. And so in general, this AB8 A-base editors have improved A-base editing activity when compared to the first generation AB7 constructs. When these deaminases are hooked up to regular wild-type SpyCas9 with its normal NGG PAM and targeted to a couple of different loci, you can see that you achieve 80 to 90% A to G conversion at some loci. And another great thing about this newer generation of A-base editors is that it has a broadened editing window, so it can actually target a larger array of potential mutations. However, interestingly, in this paper, the authors also compared these results with results where they tethered the same adenine deaminase domains to an engineered version of Cas9 and G-Cas9 that has a relaxed NG-PAM. And although this construct allowed them to target a larger population of potential targets in the genome, as you can see, the overall rates of A to G conversion are significantly lower than the same A-base editing constructs that use wild-type Cas9. And this really illustrates one of the issues that arises when engineering these base editors, and that is when you engineer for one thing, such as in a more relaxed PAM, that sometimes has deleterious effects on other aspects of the protein's function, such as overall adenosine deaminase activity. In addition to that, the relaxed PAM has the potential to increase the number of off-target editing sites at other locations in the genome, which is, of course, a safety concern for developing therapeutics. So Life Edit's sort of taken a different approach to get at to enable a broader target selection, and in such a way that avoids some of the pitfalls of engineered deaminases. And to do that, we have so Life Edit has exclusive access to the AgBiome microbial collection. So this is a huge sequence of microbial genome sequences from which we've been able to mine unique RNA-guided nucleases that have diverse PAM sequences as well. And all of these systems are amenable to different types of delivery. In addition, we've been able to identify our own unique deaminases, both C-base editors and A-base editors, that give us further flexibility in the number of targets and mutations that we can identify or that we can address. So to help illustrate how an expanded palette of PAM specificities is useful in the context of cystic fibrosis, here I'm showing you some of our lead nucleases. We call our nucleases legs or life edit genes. And for each of these nucleases, I'm showing you the number of potential guides that are capable of targeting each of these class 1 mutations. And as you can see, for each mutation, there are several guides and several systems that could potentially be used to target these mutations. And so in this instance, we're no longer limited by the PAM specificities of SpyCas9, and we can essentially target all of these mutations. Now, life edit base editors have been shown to be active in human cells. So for instance, we took our lead RNA-guided nuclease and tethered it to our first generation adenine deaminase. And we put that in hex cells along with guides that targeted several different locations in the genome. As you can see, we got pretty reasonable A to G conversion rates across several different loci. And I think it's important to emphasize that this is with our first generation A-base editor constructs. Through subsequent rounds of protein engineering, we've been able to identify adenine deaminase domains with improved activity that we're now testing in mammalian cells. So as we continue to improve our A-base editors, we're also keeping an eye towards cystic fibrosis and testing those A-base editors in the context of CFTR mutations. So for example, in this experiment, this is in human bronchial epithelial cells that were generously provided by the CF Foundation. These cells were engineered to harbor the Y122 stop mutation. And when our first generation A-base editor was combined with a guide that targeted this Y122 stop mutation, you can see that we got about up to 8% editing at one of the target A's at this location with relatively low indel formation. And while 8% may not seem like very much, it's important to remember that this is actually a level that's consistent with earlier reports of using SpyCas9 based A-base editors in this cell type with a similar delivery mechanism. So we're actually really happy with this result, and we think it's exciting, and this is a great starting point for more improvement in our A-base editors. So going forward, we're going to be improving our base editors in a couple of different ways. So one is we're doing various protein engineering approaches to improve the activity of our adenine deaminases. In conjunction with that, we're taking a target-specific approach. I'm thinking of CFTR specifically. So we're thinking about trying different promoters that have potentially increased activity in human bronchial epithelial cells. We're also trying out different nuclear localization signals. And we're thinking about different ways that we could potentially evolve our adenine deaminases to work well within the specific context of individual CFTR mutations. In addition to that, all along the way, as we identify lead guides, we're continuing to do off-target characterization to ensure that our systems have a very good specificity. And finally, once we are able to produce a robust A-base editor that is effective against a broad range of CFTR mutations, because of the flexibility of our technology, we're able to package that in several different types of delivery mechanisms. And so in the future, we hope that our systems will be able to expand the population of CF patients that we can help. And so with that, I'd like to first thank members of the LifeEdit team, in particular, Ali Crawley, who has spearheaded this project. I'd also like to thank the Cystic Fibrosis Foundation for providing extensive support. In particular, I'd like to thank Dr. Alyssa Bell and Dr. Eric Wong for providing both reagents and know-how. Thank you very much.
Hi, I'm Jonathan Gutenberg, and along with Omar Badea, today we'll be talking about how we harness novel CRISPR systems for multiple applications for genome engineering and health. Of course, we'd like to thank the organizers of the Cystic Fibrosis Conference for inviting us to speak about our work. And we hope you'll enjoy the vignettes we have on how we can apply natural diversity to develop multiple different tools with relevance to CF. So, as you know, many genomes have been sequenced. So with the growing diversity of DNA sequencing, we're starting to get a much broader picture of the variation in the population between individuals. And this has allowed us to understand many of the underlying drivers of genetic diseases, including CF. So we understand now that, of course, many of these genetic diseases can be harmful. So in CF, for example, but also in sickle cell disease or in eye degeneration, like many retinopathies. And it's important to be able to have tools that can address these differences on multiple different levels. It's also important to understand that many genetic differences can actually be beneficial. So there are many cases where genetic variation can actually lead to improvements in response to certain either internal or external factors. For example, the CCR5 delta 32 mutation in HIV allows for protection against HIV infection. ApoE mutations in Alzheimer's protect against progression of the disease. And there are many metabolic diseases with beneficial mutations as well. So understanding how and where we have the capacity to modulate nucleic acids and detect nucleic acids is important to both treat and prevent these diseases. And just to draw a little bit of a broader picture, of course, humans aren't the only organism where we have an understanding of complex genetics. So in agriculture, it's very well known that there are many different types and variations in different genes that lead to many beneficial or harmful phenotypes. So all this really comes down to the question of how can we develop tools that allow us to search and change nucleic acids? So just like we have in a Word document, a little search bar that allows us to find, detect, and replace information, how do we develop tools and how do we build our asset of tools that enables us to do that? And you can probably guess that the answer is we can go to natural diversity. So this is a picture of the tree of life as it stands in about 2016. But we like to say that, you know, we as biologists don't really build these tools, but instead we borrow them from this natural diversity. So we had a time-honored tradition of building molecular tools from naturally occurring proteins, enzymes, and other systems. So this has been going on since the 70s and earlier, with force restriction enzymes forming the basis of the original biotechnology revolution, fluorescent proteins for tracking different species in the cell, knowing how we can deliver things with viral vectors, of course, optogenetics using channelrhodopsins allows us to perturb proteins and voltage states in cells, and gene editing, which we'll be discussing today, allows us to use CRISPR nucleases to target nucleic acids, cut them, and do many other interesting things. So one thing we would like to highlight is that as we dive into the diversity of genome engineering and DNA and RNA targeting tools, it's important to remember that as we go into CRISPR, the bacterial adaptive immune system that allows us to do many of these interesting applications, typical applications of CRISPR systems would most likely involve Cas9. But the Cas9 system, which is a type 2 system of CRISPR systems, is actually only a small slice. So if you look at this tree and schematic of all the different CRISPR systems that are known, and actually it's even more broad now, you can see that Cas9 is only a small subset of this. So this really starts to beg the question, how do we discover new CRISPR systems? How do we use these new CRISPR systems to develop diagnostics and therapies? And what else might be out there? So to answer that first question, how do we use bioinformatics to find new CRISPR systems? So this is a little bit of a reframing of that earlier slide, which shows that really, despite the diversity of CRISPR systems, there's really only two classes, class 1 and class 2. And the distinction there is that the interference module, which is what people think of with CRISPR, that when we actually target a DNA or an RNA with a programmable guide RNA to cut it, in class 1 systems, that's traditionally done by a multi-protein complex, by multiple different proteins here. You can see, for example, many Cas7s in combinations with sometimes Cas10s or Cas3s. Omar will talk a little bit more about how that maybe isn't that true anymore, but generally the concept is that class 1 systems have multiple different proteins doing the work. On the other hand, class 2 systems here below involve proteins that may be much more familiar to you, like Cas9 or CPF1, which is now known as Cas12a. These proteins are a single protein, and these single effector systems actually are much easier to co-opt for taking from their bacterial or archaeal context into a mammalian cell, which is why they're so commonly used as tools. So today the question is, how to discover new single effector systems. So despite those differences, you can also see that there's similarities here. This spacer insertion module, which is known as the adaptation module. So adaptation is the process of this adaptive immune system taking on more memory, putting pieces of DNA into the genome where they can actually be used for future defense in these bacterial systems. And that's a shared system. So if we take these shared concepts and use them as anchors to bioinformatically search, we may be able to find new systems with that in mind. So what we did bioinformatically is we actually took Cas1 as a seed profile and we blasted it against, back then, about 20,000 bacterial genomes. And we found it in reference and close to other genes in the genome. We annotated some of these. So of course, if Cas1 is near a known gene, that's not as interesting, but we took these unknown genes and we actually searched for distant similarities. And we use this to actually find potential candidates, which then we can go back and blast again to break into families. And this approach actually discovered 4 new families. So they're essentially all different class 2 families, both from the type 5 and type 6 systems. These include, we refound Cas12a, this is now known as Cas12b, and Cas12c. But one interesting thing we found was actually a type 6 system, then known as C2C2, but now known as Cas13a, which actually looked like it might be RNA targeting. So we'll talk about that for a bit. So we actually characterized this enzyme, and Cas13a, as is now known, actually turned out to be an RNA-guided RNase. So it's actually produced from the locus and it's programmed by a CRISPR RNA, much like Cas9 is, but it targets RNA instead of DNA. So this, you can imagine, has many potential uses for knockdown or base editing of RNA bases, as Omar may talk about more. However, there's another interesting wrinkle that we found in this exploration, which is that it has a triggered promiscuous cleavage, which we term the collateral effect. Now, what this is if you have a CRISPR RNA and a protein and just other RNAs in solution that aren't targeted, you co-incubate them, you see essentially nothing. However, if then we redo this experiment, we add in only one other species, which is the targeted RNA, what we see is when we incubate them with Cas13a, we not only get cleavage of the target, but cleavage of other RNAs in solution. So this collateral activity, which we refer to because it cuts this other RNA that's collateral in the solution, actually is a great way to detect the presence of a RNA. So because if we see cleavage of this collateral RNA and we can detect it via its label, you can see we've labeled it here, then we can actually use that to back infer that we had a targeted RNA in solution. So this is an RNA detection modality where if we have a target sequence, we can program Cas13 target with a CRISPR RNA, and it will cleave, and we can actually detect a cleavage reporter. So what we did is we actually took this system of collateral and we coupled it with a preamplification system called RPA, but we can use many different preamplifications to create a detection system we call Sherlock. In this process, we can take a DNA or RNA, amplify it, and so that creates much more DNA using this isothermal amplification approach, which occurs at a single temperature. Then we can turn it back into RNA, and you can see that then we can plug this into that detection module. So this approach actually has many different really great properties. It can function at one temperature, 37 degrees, and is actually been shown to function at lower temperatures, almost with some work at room temperature. It can detect down to a single molecule. It can detect single base pair, resolution, specificity. So it's a very portable way to look for mutations or pathogens. And we've applied Sherlock to many different applications in viral and bacterial discrimination. It can be lyophilized for point of care. We've actually used it even to genotype patients or detect cancer-related mutations in circulating DNA. So it's a very broad platform that is of general diagnostic use. Of course, you can imagine that, especially with our current ongoing infectious disease pandemic, that there are many applications, and we'll touch on those today. So how do we use Sherlock for COVID detection? So Omar and I are actually co-founders of a company that is commercializing Sherlock, and about a year and a half ago, I would say now, we actually got a emergency use authorization for the use of Sherlock for COVID-19 tests. So this is the first emergency use authorization or FDA authorization of any use of CRISPR, but it was actually the generation of using the Sherlock platform for detection of COVID. However, that authorization was actually only for high complexity situations. So in collaboration with Feng Zhang, our old mentor at the Broad Institute, we actually worked on developing a system for portable testing and detection of COVID-19. So we developed a system we call Stop COVID, using actually a Cas12 enzyme, which also has collateral, but it allows us to do very robust high temperature amplifications, where we can simply dip a swab in an extraction buffer. We can isolate the nucleic acids on magnetic beads. We can add a Stop COVID mixture, which includes the CRISPR enzymes and the isothermal amplification mixtures. We can heat that, and we can rapidly detect either on lateral flow strips or with a fluorescent reader. So this makes it very easy and very simple to do point of care detection. So how does this approach compare to other diagnostic approaches out there using CRISPR? So we actually, in February of 2020, released a protocol, which we termed Sherlock COVID, that we used for detection of SARS-CoV-2. And the Sherlock Biosciences authorized detection workflow, as well as a Mammoth Biosciences detection workflow, have all been applied for COVID detection. However, they all involve a step, which is signified by a red arrow here, of opening the tube, which increases complexity. The Stop COVID approach involves a rapid extraction and a single step incubation that allows us to both reduce the complexity and increase the speed of the reaction. So in collaboration with groups at the University of Washington, we actually tested this on a panel of 400 patient samples. And we actually found that not only could we detect these patient samples across a large range of CT values for qPCR, we actually found that our time to threshold correlated with the CT value. So higher CT values and lower abundance samples took longer to detect as well. So across this panel, we actually achieved very high sensitivity and specificity. So 93% sensitivity and 98% specificity on these patient nasopharyngeal swab samples. And we view this as a great example of the ability to do point of care detection using these CRISPR enzymes. So this is only one example of the many applications of these CRISPR systems and how we can use them for diagnostics like Sherlock. We're also working on increasing the multiplexing capabilities. We can use it for quantitation. Of course, as you saw, we can use it for lateral flow detection. And most importantly, we're working now to develop this into a portable diagnostic. So thank you very much for listening to this application of our new CRISPR systems. And now Omar can discuss how we're using these to do new types of targeting of genetic disease in mammalian cells.
Great. Thanks, Jonathan, for that great introduction to sort of our new work in new CRISPR enzymes and other applications for diagnostics. Now I'm going to dive into their applications inside cells and a lot of the relevance of these tools for cystic fibrosis. Just to dive in, there's obviously a lot of layers of biology inside the cell that RNA is involved in, and it would be great to have tools to manipulate these RNAs and their interactions with DNA. And so we've spent a lot of time developing a suite of tools that can be involved with modulating RNA, including RNA knockdown, base editing, and even modulation of translation or splicing, some of these applications which would have a lot of relevance for cystic fibrosis. Now, to sort of just briefly touch on knockdown, a lot of these Cas13 enzymes can actually be reconstituted inside cells to target RNAs, and you can target them towards reporter transcripts like GLUC and show that we can actually get knockdown up to 75% or so. When compared to shRNA, it's actually as good or if not better in some cases. Cas13 is also great because it has quite exquisite specificity. So if you look at total RNA in the cell, the black distribution is RNAi and the red distribution is Cas13. And you can see that the sort of RNA distribution inside the cell is much tighter, except for the transcript being knocked down, showing better specificity. And that's the case for both Cas13A and Cas13B. Now, there's been issues with Cas13 in that people have started to see that the collateral effect, which Jonathan described for diagnostics, can also happen inside cells. And what you end up seeing is that cells that have targeting can have reduced cell growth and actually loss of RNA integrity, where you can actually see total RNA to start being degraded, including ribosomal RNAs. So, we've spent a lot of time trying to figure out, can we build better RNA targeting tools on top of these enzymes, or even find newer enzymes? And so just to dive into that a little bit, obviously we talked about the CRISPR universe and how there's many different systems and signatures of these systems. And if you start to mine even more, you can find more flavors. And recently Eugene's group showed that there are some flavors in the type 3 world, which are actually these complicated, multi-subunit systems. You can see a lot of genes here. But there was one system shown here where actually a lot of these enzymes had consolidated into one larger enzyme, as shown here. And it looked like it targeted RNA because of its domains. And so we were pretty intrigued by this enzyme. So we reconstituted it in our lab. And what we found is that compared to Cas13, as shown here, there was no collateral effect with the enzyme. So Cas13 has this nice curve, which we can use for diagnostics. Cas11 doesn't have that when targeting its target. However, also in cells, Cas13 would cause significant growth reduction, as shown here, but Cas11 had no effect on the cell growth. Now, when you look at its ability to knock down RNA in the cell, it's still as good as Cas13, if not better, as in the case of Cas13A. It's almost as good as Cas13D. So we were quite excited that there actually can be these systems where you could get programmable RNA targeting, but not have the toxic side effects of Cas13. And if we dive into this a little bit more, of course, in HEC293s, as we've shown previously, you don't really see growth defects from RNA targeting. But in other cell lines like U87s, you can do knockdown and actually see growth defects. And so shown here, in mammalian cells, you can see Cas13 has a significant hit on cell viability, although it does get knocked down, whereas Cas11 can still get more than 50% knockdown, but not take this hit on cell viability. So another technology we wanted to talk about was base editing, which we've done with catalytically dead Cas13 variants. This can be used, of course, for modulating amino acids of proteins, correcting protein mutations, even modulating splicing, which again would be relevant for CF. So a question we often get is, why do RNA-based editing when there's DNA editing? And I would say the main answer there is you may want to do temporal modulation so that you don't have the permanent sort of effects of gene editing if you're concerned about targets or don't know the long-term effects of the change you're making. So, and this can be used, of course, from temporally modulating protein function, correction of disease, temporally modulating splicing, and even correction of Mendelian disease. I think a really big application of temporal modulation would, of course, also be for things where you might want to regenerate cells post-injury, modulate the immune system, modulate pain, modulate, infectious disease, like treat SARS-CoV-2, for example. There's a lot of applications where you might want to do this. And how we do RNA editing is we actually use Cas13 to recruit endogenous enzymes that normally convert adenosine to inosine, which is read out as a G and gives you functioning A to G change. So what we're doing is we're basically building on this Cas13 system to fuse an ADAR deaminase domain to it, and then we can use a guide to basically target an RNA transcript. And the guide will make the ideal RNA duplex that an ADAR would edit, and we can actually make the exact mismatch across from the A that ADARs prefer. They actually prefer to edit within a mismatch bubble. So by doing this, we can actually program the ADAR to precisely edit where we desire and get an A to G change as a result. And we've called this technology repair. Now, by modulating different parameters of the guide, including the length and the position of the A and the mismatch, we actually get much different editing rates. And as shown here, we've seen up to 30% programmable editing using this repair system by being able to do this. Now, of course, you can see there's a lot of off targets in the duplex region because the ADAR will also edit other regions. And so we characterized off targets in the genome and found that the original repair system, while it could get 90% on target editing as shown in orange here, it still had issues with off targets throughout the rest of the transcriptome. So we went back to the drawing board and actually tried to make a mutagenized version of repair shown on the bottom called repair v2 that actually had way fewer off targets, and it would slightly reduce the on target rate, but it actually made the system almost 1,000-fold more specific. And just to drill this point home more, you can see the reads around the target site here where there's a lot of G's being created around the target site. And the more specific version is actually quite good at just hitting the on-target site. And so by just mutagenizing the protein to sort of destabilize the target region a tiny bit, we can improve this editing rate, which is really useful for doing programmable and temporal editing. So, of course, repair is quite powerful doing different changes. And as shown here, it'd be great if we could actually do CDU changes, because these are if you could do CDU deamination, and there's many more sites you could do for phosphorylation, catalysis, and other post-translational modification modulations. And we actually ended up having a paper on this where we directly evolved a ADAR to do CDU rather than A to I mutagenesis, and we're able to get basically more base edits in the RNA toolbox as well. And so we think, having a robust RNA toolbox is quite useful, and some of these tools might be useful for the cystic fibrosis community as well. So, we're pretty excited that we can continue to find new RNA effectors that are safer for cells and could be used for all sorts of RNA therapeutics, as shown here, where there are a lot of different functions you may want to do. I think with relevance for cystic fibrosis, of course, is modulation of splicing to maybe skip certain exons that are problematic, and that's actually an area where people have shown Cas13 could work, and Cas11 should also have similar effects as well. And so finding better and safer enzymes that can work inside cells is, I think, really important for deploying these things as therapeutics. And so just build off of that, we're continuing to build on this RNA toolbox and trying to find more tools to fill this toolbox with. I think one thing we want to close on as kind of a teaser for where we think the field is going is, we obviously have had, nucleases that can do programmable cutting. We've done a lot with base editors to do programmable, specific base changes. Then we sort of found that there were these, transposases and recombinases that could associate with Cas enzymes, although those have not functioned well in human cells yet. And then, of course, prime editors, which can do most small changes in the genome. However, what's still lacking amongst these tools is a way in mammalian cells to be able to basically do large insertions. So if you look at all the types of changes we can do now with different flavors of editors, most everything is covered. I would say large deletions is probably covered with multiple guides and making cuts, or even sometimes you can get big deletions with a single cut. But large insertions is not really covered by prime editing, and you can kind of do it with HTR, non-homologous end joining, but it doesn't work as well inside cells, and there's of course caveats. And as I mentioned before, Cas recombinases and transposases are also not as active in mammalian cells because of their multi-subunits and difficult to reconstitute in the genome. So, we're obviously very interested in this space, and I think our group has obviously been working on programmable integration tools that can hopefully work. And the hope there is we can eventually switch to a therapeutic approach versus fibrosis, where we can just pop the fibrosis gene directly in the genome at the endogenous site. And the appeal there, of course, is all these other approaches are so specific. You have to develop a drug for every mutation of cystic fibrosis, and with more than 1,800 mutations, you have 1,800 drugs. And so we were really excited about this idea of just endogenous gene replacement, where you can have a one and done therapy that applies for everyone and treats all mutations, regardless of where they are. So I think just to highlight, there's a lot that's been done in the DNA targeting space, and I think we're almost at the frontier of being able to complete this toolbox. So, as I mentioned before, we gather a lot of our fodder for the toolbox per se from natural diversity. We screen all sorts of prokaryotic and karyotic genomes to find these enzymes and try developing them as tools. And we're continuing to do this, including for the gene integration stuff I'm talking about, and also hopefully for other challenges like gene delivery and other things to really complete the gene therapy toolbox. So on that, I'll just say, as we discussed today, there's a lot of exciting work for RNA modulation and being able to develop tools that are safer and useful for RNA therapeutics. And, I briefly touched upon, I think the exciting new work that's going on in programmable gene integration, and hopefully we'll see come out soon. And I think in general, combining these technologies for a really robust cell engineering toolbox is really exciting and something, we're obviously working towards. So on that note, I'll say thank you to our lab. And of course, as always, we're hiring and recruiting. So if anyone's interested in building the next generation molecular tools, that'd be really great. We actually just got a grant from the Cystic Fibrosis Foundation to apply some of our, DNA and RNA targeting technologies towards CF. And so I think there's a lot of exciting opportunities in this space if you're excited. So shoot us a line. And of course, all the great, people in our lab that have worked on these technologies, as well as our collaborators on different approaches that we talked about. So thank you again.
Hi, I'm Paul McCrae from the University of Iowa, and on behalf of my co-chair, Hillary Valley, and myself, we would like to welcome you to the symposium on gene editing to target CFTR DNA repair. We'd like to thank the presenters for the talks that you just heard, and we look forward to a lively discussion. Before we start, we're going to ask each speaker to introduce themselves briefly, and we will go in the order of the presentations. So first is Dr. Amy Ryan.
Hi everyone, I'm Amy Ryan. I know in my presentation it said I was from USC, but I have moved very, very recently to the University of Iowa, where I'm now an associate professor of anatomy and cell biology.
Great. Dr. Greg Newby.
Hi, I'm Greg. I'm a postdoctoral fellow at the lab of David Liu at the Broad Institute, and currently on the job market for faculty positions too.
And Dr. Philip Borden.
I'm Phil Borden. I'm the Associate Director of Gene Editing at Life Edit Therapeutics here in Morrisville, North Carolina.
And finally, we have Omar Abudeye.
Yeah, hi, I'm Omar. I run a genome engineering lab with Jonathan Gutenberg, who also shared the talk, but is not present for the Q&A session. And yeah, happy to chat about some of the stuff we presented.
Great. Well, we'd like to keep the discussion kind of free flowing, but we do have some questions from the audience, and we'll get started with that. Feel free to ask us questions as we go along, and we hope we'll have a lively discussion. So I wanted to start out with a question for Amy. So this is, to what extent can we ascertain whether static mucus is altering differentiation? What is the time course of these events? And is altered differentiation dependent of the mucus barrier changing the susceptibility to nucleotide transfer or gene editing delivery?
That's a tricky question to answer. I'm not sure we actually have the answer to that right now, but I think it's a very good question and something that is highly likely to have a significant impact on gene editing, gene delivery. But yeah, I don't think I have a really efficient answer to that. But I think definitely the mucus and the composition of mucus is going to change the dynamics of cell differentiation and the cellular composition in the airways, which is therefore is going to have an impact on delivery by whichever mechanism we're delivering.
Okay, I'll send another one your way, just see how this one goes. So how feasible do you think it is to edit the myoepithelial cells given their location and regeneration only in response to severe injury?
Yeah, that's another good question. Again, I think it's going to be challenging. I think targeting the epithelium efficiently is going to be challenging, however we look at it, because we really need to access the stem cells, and the stem cells are the least accessible cells in the airways. Targeting from the apical surface. The other way of thinking about this is maybe targeting from the basolateral surface through the blood, or in a different way, maybe utilizing cells that are able to migrate and infiltrate that basolateral surface. Maybe a different way of thinking about this rather than going in from the apical surface. But yeah, myoepithelial cells, because of the location in the submucosal gland, which is likely plugged with mucus and CF, is probably going to be one of the more challenging places to target.
Thanks, Amy. So next up, I think we'll have another question related to kind of the cell types and what cells we want to edit. So I'm going to direct this question first at Greg, but Phil or Omar, feel free to jump in as well. And the question is, do base editor strategies require cell division for mediating correction? If yes, how do you think these strategies can be used to correct the mutant CFTR in fully differentiated airway cells and or in slow proliferating basal cells?
Thanks. Yeah, I think one of the really nice things about base editing is that it really doesn't require cell division for activity. One of the results I was most excited about from last year was a paper by John Levy in our group, and they observed that they could get over 60% of total alleles in the mouse cortex to be edited, which is generally not a dividing organ. And so we can get really, really efficient editing even in non-dividing cells. And so hopefully we won't be limited to one cell type or another based on their division rate. However, the delivery problem, I think, is still one of the most substantial ones now that we have good editors for all these mutations, and figuring out how to get it into the necessary cell types to durably correct the disease is the next big challenge, I think.
Thanks, Greg. I'm going to send one more your way about base editing, specifically where we're talking about base editing, and that's about bystander effects. So could you comment on any bystander effects that you might have seen for the 1282X mutation, so bystander effects on the other allele, and can you also comment on any that you might have seen for the R553X edit?
Yeah, the bystander, the propensity of base editors to edit bystander nucleotides changes based on the activity of the deaminase quite a lot, and based on the target site and how nearby other nucleotides are. So the 553X target site was particularly clean one. There aren't any nearby adenines that would lead to a non-synonymous change, and so we could very cleanly edit only our target site. 1282X, that's not the case. There's an adjacent adenine that would cause a non-synonymous change, which we're not 100% sure yet, but has been reported in a few cystic fibrosis cases, so we think it's possible that it's deleterious. So I think it'll be a question of really fine-tuning the deaminase to target only our targeted nucleotide that we want to hit to correct the disease. We're probably not at the place yet where we can say we're really optimized to do that at 1282, but our current generation editors, I think, at the 553 site are going to be much better.
think while we're talking about base editing, maybe Phil, if you could answer this one about Y122X base editing. So the question is that A to G editing will only correct Y122X to Y122W. Does the W, the tryptophan, restore CFTR function? So have you looked at function in these cells in addition to the editing?
Sure, that's a great question and is relevant to many loci depending upon exactly what the edit that we can make is. I think the answer is that no, we haven't looked at that, but that's certainly something, a challenge that we would have to overcome in the future and something that we would want to be keeping in mind.
I'd like to go back to the talk that Amy gave regarding stem and progenitor cells. And one focus of this work is to develop technology so that cells could be edited ex vivo and then returned to the lung. I wondered if you could comment on what criteria should be used to characterize an iPSC differentiated cell as being suitable for a donor cell in a translational sense.
Yeah, so this is something that's a constant discussion in the field is what is going to be the defining criteria of that iPSC-derived stem cell or basal cell to classify it as equivalent to an endogenous stem cell. I think it's still a big question. I mean, the criteria would be that it is equally capable of differentiating as an endogenous stem cell. How similar transcriptomically it's going to need to be, it's still, I think, a debate, how similar do you need to be, or does it just need to be able to functionally perform the same? I think if you've got a cell that's able to functionally perform the same, then I think you're fulfilling some of the main criteria there. But then you need to know that, long term, do those transcriptomic differences make a difference? So I think these are some of the questions that are really important to be answering as we move closer towards that now. Now we've been able to differentiate iPSC successfully to airway cells. With a lot of the studies right now starting to focus on transcriptomics, but then we also had the epigenetic status of these cells and to know how significantly that is impacting their performance as well, because that's going to be significantly different in the CF lung to a normal healthy lung as well. So I think that's a great question, something that we need to now move towards defining those criteria of what it means to be an iPSC-derived airway cell and how similar they actually need to be. I think that's the stage we're at in the differentiation process right now.
Another question related to your talk, you mentioned how inflammation or inflammatory cell types could influence the pluripotency of stem cells or progenitor cells. When you think about this in terms of whether it was a in vivo therapy or an ex vivo therapy, how are we going to know what the optimal timing is for these cells that are patient derived? Or it seems like there's a lot to learn there. Do you think there are specific inflammatory cells that are particularly important in driving these effects on stem cell capacity? You mentioned macrophages, for example, in your talk.
Yeah, you're not asking easy questions here. I mean, we're at the beginning, I think, of really understanding how those cells are influencing the stem cells and the stem cells' behavior. I presented preliminary work from our lab on the macrophages, and they definitely seem to have a significant impact on the timeline of differentiation of those stem cells. Whether it ultimately is change in differentiation, we don't really know that yet. But it's highly likely it is. And therefore, as you say, when you're thinking of the disease and disease progression in CF, then is there an optimal time point before you get a significant buildup of these inflammatory cells in the proximity to the airways? Is there a good timeline? And I think it's a good question that has yet to be answered. I think we need to go in and look at that. The other cells would be the neutrophils. We've just done some work on COVID and looking at neutrophils and how neutrophils are influencing the airway repair there. And again, we've seen a significant impact of the neutrophils on that process. And we know that neutrophilia is also rich in cystic fibrosis in proximity to the airways. So I think both those cell types are likely to have impact. And whether there's a timeline you need to get in before you establish chronic inflammation in the airways, I think we still need to look into that for sure. But I think we need a good in vivo model to do that. In vitro is only going to tell you so much. I don't think you can put a price on doing that in vivo.
Yeah, it's really a question that applies to lots of therapies. And I think we probably all share the perspective that if we had something that was efficacious, we'd like to do it as soon as possible to
absolutely
prevent disease advancement.
We've had a couple of late breaking questions, including one for Omar in particular. And I think maybe Greg, you could weigh in on this a little bit as well, which is how efficient have your attempts been to prime edit CFTR or edit CFTR in general? Is there any issues with low efficiency due to sort of unique features of the CFTR gene, specifically that it tends to be very AT rich? So maybe Omar, if you could start with any editing in CFTR, and Greg, maybe if you could jump in after that.
Yeah, that'd be great. I mean, I think we haven't done too much in the RNA targeting space for CFTR. Although I would imagine, with Cas13s and now Cas711, we haven't seen issues necessarily with binding in the RNA for AT-rich regions. I would imagine you could use, these systems for either modulating splicing and binding, and that could probably work fine because the splicing applications are quite robust. I will say in the domain of gene editing, we've done a decent amount of this. Like we just put out our new technology called PASTE for doing programmable gene integration. And, one of those areas we are looking to is CF, especially since we just got a CF grant to do this. And I will say the CF region in the DNA has been harder to target. So we've actually been taking a lot of effort to screen, introns and upstream of the gene, and editing does seem to be a lot lower. I will say, I think we're starting to see, editing in the range of 10 to 20% for being able to use sort of our integration system. But I will say in our experience, it's been hard. So, yeah.
Thanks, Omar. Can you tell us a little bit more about PASTE? I know you have a recent bioRxiv paper out, but in case not everyone has seen that, can you expand on that technology a little bit, which is really cool?
Yeah, happy to. So it's been a long challenge, I think, in the gene editing field to be able to do programmable gene integration. I think obviously it's possible with repair pathways like non-homologous end joining and HDR, but especially with HDR, that doesn't work when you go into the body with a lot of non-cycling cells. And so we've been wanting to solve this problem for a long time with transposases and integrases. And we realized, we could leverage some of the, work, for example, like David Liu has done with prime editing, to be able to finally marry sort of programmable placement of beacons for integrases that are hard to program with efficient integrases that actually can insert, sometimes up to 70-80% in human cells and mammalian cells. And so we released this program where we show a single protein construct that basically can be guided by a guide RNA to insert anywhere you want in the genome, and we show they can work in non-cycling and non-dividing cells. And we're pretty excited for therapeutics. And one thing I will say around this is I think the gene editing field a lot of times has been trying to get more and more precise, like, what's the exact, base edit we can make or the exact small change we can make. And the problem you hit actually is a lot of diseases have multiple mutations, right? So like, let's use CFTR as an example. There can be thousands of mutations. And so it's hard for a company to say like, I'm gonna make 1,800 gene editing drugs. But with a gene integration approach, you could have a one therapy that's permanent for all patients with just a single sort of dose. And I think that's really the vision here with gene integration, is that we can give it all patients a treatment. So.
Great, thanks, Omar. Super exciting technology, and the gene editing field in general is advancing so rapidly. Base editing, prime editing, PASTE. It's really cool to see all the new technologies that are coming out, and yeah, very, very neat. And it's really cool to see too, especially some of these new technologies that are coming out, like you said, for large integrations, which I think particularly for CF is going to be really important. Yeah, Greg, do you have anything that you wanted to follow up on specifically about editing in CFTR? Any additional insights to add in that area?
Yeah, I'll just jump in with our experience as well. We've certainly tried hard to conduct prime editing to correct cystic fibrosis mutations, and it's been more challenging than in pretty much any other site in the genome that we've worked on. We're still trying to work out why that is, whether it's AT-rich, could be could be one factor, but base editing still does work well. So we don't think it's about Cas9 accessibility or protospacer binding. So yeah, that's definitely something we're still looking into. Our lab recently published 2 papers about improvements to the prime editor system that I spoke about in the talk. So we're hopeful that some of those will help the application of prime editing there.
It seems like a common theme in the evolution of these technologies is mining microbial genomes to find the next best thing. I wondered, Phil, if you could talk about this. You referred to the database that you guys have used in your technology development, and of course, other labs, Eugene Koonin, Jennifer Doudna and colleagues have, also spoke of mining, these such databases as Omar did. What's different about the database that you're working with? Is it complementary or similar to what other people are using? Could you comment on your approach?
Sure, I would say it's complementary. So AgBiome has done a really good job of trying to sample a diversified set of microbes from around the world. And so I think that gives us access to a diversity of potential gene editing tools as well. And one of the advantages of the AgBiome collection is that they specifically go after non-pathogenic bacteria. So we have some hope that the systems that we're mining out from there are non-pathogenic from the get-go, which has significant advantages when in terms of thinking about a human therapeutic.
In a follow-up to that, one of the challenges people report with mining bacteria for new editing enzymes is the low rate at which these enzymes might perform in mammalian systems. How effective are the things you're discovering for working in mammalian cells?
Sure. Yeah. I mean, we face that complexity as well. But as part of our pipeline, we determine what enzymes and systems will actually work in mammalian cells kind of at an early stage. And so that helps us to winnow out things that aren't really worth moving forward with.
Thanks.
So we've talked a bit about editing technologies, and I just want to shift gears a little bit for a second and talk about delivery. Obviously, delivery is critically important. And so one of our audience questions was, how do you propose to best administer the new gene transfer and editing vectors effectively to inflamed, injured airways? So I know this is a big topic for discussion, but if anyone wants to kind of jump in here.
So I guess I can start from the perspective of working for a therapeutics company. And I would say that delivery is probably the #1 challenge that we face. And being able to harness new delivery technologies, such as things like cell penetrating peptides, things like that might give us hope in the future for being able to deliver these gene editing systems that work really well.
Yeah, I think also we've seen nice reports of using lipid nanoparticles to target the lung, but it's unclear still whether those target mostly endothelium only or epithelium or cells that can differentiate into cells that form the epithelium. And so figuring that out will be important, as well as whether we can deliver through the bloodstream or through some sort of inhaling nanoparticle system. Just working out the best tools to deliver into those cells.
So can you tell us a little bit more about the size of your legs? So are any of those significantly smaller than the current ABEs, and kind of what are the delivery options for those therapeutically? For example, would they fit in an AAV? You talked about, we've talked about lipid nanoparticles. Can you comment on the size of those?
Sure. So that's kind of something else that we filter, is another filter on the systems that we bring to the platform, is we really want to move forward with things that are significantly smaller than Cas9 so that we can deploy them in, in an AAV, even in a full base editor form, or that are amenable for LNP delivery or other mechanisms.
Omar, in your work, you guys mentioned Sherlock, and one of the hallmarks of cystic fibrosis is colonization with microbes. Mainly, people think about bacteria. Do you think that could be any role for this detection system for detection or identification of different species of bacteria or specific subsets of a certain bacteria?
Yeah, absolutely. I mean, I think, we've been working very hard to make these, Cas12 and Cas13 based diagnostic approaches to be very specific. And in fact, we have demonstrated that single mismatches or even deletions can be detected using this approach. And so, you can think about using this to, if you really need to know the species of bacteria, we'll go after like ribosomal RNAs. You could use this for going after like antibiotic resistant mutations, which we've actually shown a little bit of in sort of like the Klebsiella sort of world. And yeah, I mean, we've also done genotyping. So the specificity is definitely there to be able to do that.
Speaking a little bit more about inflammation and bacterial infection, Amy, in your talk, you were talking a little bit about CF and non-CF macrophages in the epithelium, and that data was very interesting. Could you provide a little more detail about the origin of the macrophages? How long were they in culture after isolation? Is it possible that they were remembering inflammation, or had they been in culture for a while? And could you also comment on the genotype of the macrophages? And I think this is relevant because there's been some discussion about, direct interactions of CFTR with other proteins in the membrane. And so, if it was something like a G542X genotype, then there would be little to no CFTR present. But if it's something like F508del or G551D, then maybe some of those protein-protein interactions could still be there. So could you comment a little bit more on the details of that study?
Absolutely, yeah. So the work we've done so far, which unfortunately got put on hold due to COVID, so most of it was before then, and we haven't gone back to readdressing it, but we isolated the macrophage using a unique fast isolation that my postdoc established. So within 2 hours, we have macrophages from explant lungs. All the macrophages that we've analyzed so far were all from delta F508 lungs, so we haven't had any comparison with other mutations. So that answered part of your question, I think. So I definitely think that the mutations in CFTR are impacting macrophage activity. We did, to control for variation between patient to patients, we did all our initial experiments using the same cell line from patients. To look at the interactions. The only differences were the macrophages from the healthy lung and the CF lungs. Obviously, when you're using patient-derived cells, there can be a lot of differences patient to patient. So we're trying to minimize those while we're studying the macrophages. And the interactions with the macrophages and the epithelium is significantly different to when you look at epithelium alone or when you look at macrophages alone. The activity of both parts of that equation are changed once you have a co-culture system established. So we still are right at the beginning of kind of our work there, really looking at what it means for these mutant macrophages from CFTR to be studied with mutant epithelium or control epithelium. So there's not much more I can say on how they interact yet, but I think it's important to consider that CFTR is expressed in more than just the epithelium and may have a significant impact on the function of those other immune cells as well. So yeah, I mean, when we're thinking of gene therapy, we're only looking right now at correcting the epithelium. So still having mutant immune cells present is going to impact that epithelium. It's something that may need to be considered as we move forward with gene editing therapies.
I had a question for Greg that maybe Omar may have some thoughts on too. So it seems with the prime editing approach, you're showing multiple stepwise improvements with, from a variety of angles. These also seem to be perhaps increasing the size of the cargoes you might need to deliver to be effective or be more efficient. But, just drilling down on one of the examples that you talked about ways to modify the PEG RNAs, and you mentioned this MP-NOT. Is this a commercially available approach? Is this something that could be engineered into an expression vector, or would it only apply to RNP delivery? Could you tell us a little more about that?
Yeah, absolutely. Thank you. I think that the MP0 and Evo pre-Q1 are these two stabilizing motifs that we find give substantial boosts to editing efficiency, especially in primary cells where the nucleases may be more active. And I think that those are of the most recent improvements to prime editing. Those are the ones that are most accessible to all of the different formats, because those motifs are very short. They're only 30 to 45 nucleotides or so. And so that's not so much space that it'll stop you from encoding it in any viral vector. And you can also, well, synthesizing synthetic pegRNAs for uses with RNPs is still a challenge, and we can't make those unlimited length, or even as long as we'd like them to be right now. But for shorter pegRNAs, it's still possible to fit that motif on and make a synthetic RNA molecule. So we think that these motifs will help pretty much any context that you'd want to deliver a prime editor in. One of the other improvements being the MLH1 dominant negative domain that transiently inhibits mismatch repair. That would be a little bit more challenging because that is a several hundred base pair or KB gene that you'd need to co-deliver. So that would be harder for an AAV approach, but might still be suitable for a nanoparticle where you're delivering mRNA.
So, and have you tried to deliver the MLH1 dominant negative in protein form?
We haven't purified proteins of that, but we can transcribe mRNAs of both prime editors and MLH1, and we saw that did help in the primary T cell example that I showed. I believe we used mRNA for both prime editors and MLH1 dominant negative there. Yeah.
So we've
Sorry, Omar, any thoughts on, with your base system that's evolving and how this would apply?
So I will say, we've played around with the pseudoknots and like stabilizing domains for our longer ATG RNAs, which have the attachment site built into the PEG RNAs. I will say we haven't seen a huge improvement in cell lines. I do think if you're delivering DNA expression into primary cells, I would expect that to actually boost based on Greg's data. But the other thing I'm curious about, we played around a little bit with synthetic guides, and I'm not sure that approach necessarily work for synthetic guides that are like modified to prevent from nuclease degradation. Maybe curious to hear Greg's thoughts on that, because we do a lot of synthetic guide work and see pretty good editing with them in different cell types. But we haven't tried adding the pseudoknot because they'll get way too long. They're already like 180 base pairs with the attachment site, which is like really near the limit of RNA synthesis. But I don't know, I'd be curious to hear your opinion, Greg, on like why the pseudoknot would help when you already have like degradation resistant synthetic guides.
Yeah, I think that we would expect the effect to be lower if you already make a synthetic guide RNA. It's most common to include phosphorothioate and 2'-O-methyl modifications on the 3' end that will prevent the nucleases from degrading it. But we think that that's not a perfect solution, and that nucleases will still eventually degrade those PEG RNAs. And so having the motifs in our hands did actually still improve their activity. And if you're already using a very long PEG RNA of 180 nucleotides, then I haven't found a company yet that will synthesize one that's that size with our motifs on it. But some PEG RNAs are only 120 nucleotides, and in that case, then you still have space to add those 30 nucleotide motifs on.
That's great.
So we were just talking about prime editing version 5. There was some discussion earlier about base editing version 10, and I think everyone expected all of these editing technologies to advance and evolve. How much room do you think there is for continued development and evolution of these technologies? Obviously, there's new ones that are being developed, but evolution in this space, like, are we hitting the plateau? Is there more space to be explored here with PAMs and sizes and alternative enzymes? And so, Phil, maybe we'll start with you, and then anyone else wants to jump in after that.
Sure. I think there's a lot of room for improvement in this space. The thing about base editing is it's very target specific. So depending upon where you're trying to look in the genome, you may have a system, one A base editor works really well at one locus, maybe it doesn't work so well at another locus. So I think that there's a lot of room for improving both the overall efficiency of base editors so that they can be more robust across any place that you choose in the genome, and also to improve their specificity at the same time, keeping their specificity to prevent off-target editing.
Yeah, I agree. Along those lines, I think so for C base editors, The original BE3 and 4 that our lab published have very poor activity at a GC motif, and recently there were EA3A base editors that have different context specificities, and so I think that one really exciting avenue to help improve A base editing and its both its activity at the target site and its specificity would be to select for a really specific context for the mutations you're targeting, and then yeah, you hopefully get both of those effects at once, both increasing your desired editing and then eliminating other edits throughout the genome. I was also curious to maybe Omar or Philip could comment on it, but I know that we've been exploring a lot of different bacterial sources for these sorts of enzymes, but is there any estimation of how deep of that surface we've scratched if we've looked at more than half of what we think is out there, or if we're really at like 1% and 99% of either deaminases or CRISPR-associated enzymes are yet to be tested?
Yeah, I mean, yeah, I'll speak on, I guess, our side. I think, we're barely scratched the surface. I mean, we put together 10 terabytes worth of metagenomes and genomes. It's just like, there's just so much out there, defense systems, different types of enzyme domains, that are clustered near CRISPR-Ras, and also clustered other stuff we're interested in. And in general, I think I'm really curious, what are maybe general classes of RNA-guided nucleases, like Fung, recently published ancestral CRISPR proteins. I think that kind of hints that, maybe a lot more things could be RNA guided than we expect. It's not just CRISPR. And I think, pulling on that thread is also really exciting, and, more work will be done in the next few years. So.
And I totally agree. That's a question that we ask us all the time is, how we mind our collection for everything out there. And I think the answer is going to be no. There's going to continue to be things that are interesting that can be harnessed to develop therapeutics.
Yeah, I think it's important to remind ourselves that this field sort of appeared about less than 5 years ago. So I would hope we're not at the end of it, that there's a long way to go and new things to discover.
We have a couple more audience questions that I want to make sure that we get to. So maybe if we could have some, we're getting low on time, some short answers here. So the first one I'll direct towards Omar. And so that's what is the relative advantage of inserting a large gene integration strategy via something like CRISPR or PASTE or something like that versus a mature CFTR mRNA without editing? So can you just kind of generally comment on what you see as the advantages of that versus like an mRNA therapy?
Yeah, I think that's an interesting question. Obviously, in the mRNA space, I'm most familiar with Recode and Dan Siegert's, work trying to deliver to the lung. And, we've actually chatted with Dan on and off about it. I mean, I think, my impression of mRNA therapeutics is you have to dose and you have to figure out what the dosing is, and someone would have to take it forever. There seems to be a school of thoughts that for certain tissues, maybe if you express a protein like a CFTR receptor in the cell surface, it might actually stick around and then the protein will not degrade that fast. So it's not dosing, every week or every couple of weeks that you might expect. I haven't seen necessarily the data on this, so I think that's an evolving story, whether that's possible or not. But obviously, in lieu of actually seeing how well an mRNA therapeutic works in humans, I mean, I think intuitively a gene editing approach makes a lot more sense. You just have to dose once, it's permanent, hopefully their cells express the protein forever and you don't see loss, especially with what we're trying to do, which is programmable integration at the endogenous locus under the endogenous promoter. So you don't get loss of an exogenous promoter like an EF1 alpha or something with time. I think the challenge though is delivery is hard. I guess that's the theme of this Q&A session, right? And getting through the mucus and getting to the basal stem cells that you need to get permanent editing, right? Because if you just hit the epithelial cells, they'll eventually turn over as well. And so I think we obviously need better delivery to the right exact cell types as well. And I don't know if it's quite there yet, but obviously we're excited to look into it. Yeah.
Thanks, Omar. Maybe we'll do one more question, Greg, and I think this one is for you. So this one's about multi-subunit CRISPR types. And if we're able to use them as separate proteins, which you could then simultaneously target to a target locus to reduce off-target effects or to help with delivery, can you comment on whether or not this has been done for base editors and kind of where you potentially see that going?
Yeah, definitely. I haven't heard of that for specific nuclease, actual CRISPR-associated proteins, but for deaminases that we use for base editors, there have been some reports where you can split that deaminase into two pieces and then target them individually to the desired site with two different Cas enzymes. And then only at that target site then would they be able to reconstitute and actually act. So this could increase their activity at the on-target. Sorry, it wouldn't necessarily increase their activity because it's a harder problem now. You're asking two pieces to get there at the same time, but it should substantially decrease their activity elsewhere in the genome because now it's going to be very rare where you have those two pieces at the same time, the same place.
Well, on behalf of Hillary and myself, we'd like to thank our speakers for excellent talks and the audience for pushing questions forward. I think we've had a lively discussion, and we thank everybody for participating in this session.